Very cool. Hi everyone, and welcome to this lecture on Chapter 5. So this should also be a two-part lecture. So we'll go into the first half of the chapter here. So in Chapter 5, we're going to be talking about lipids. Lipids are the second of the three macronutrients that we're going to be talking about. So we have carbohydrates, fats, and proteins. Those are our three macronutrients. Lipids and fats, I use that word synonymously. There is a subtle difference to the two, but for our purposes, we'll think of lipids and fats somewhat synonymously. So lipids is kind of, I like to say, the umbrella term. Lipids it's a large group of molecules. There are many different types of molecules, lipid molecules. And a specific defining characteristic of lipids is that they are insoluble in water. Where did we see this word insoluble before? We saw that when we talked about insoluble fibers. So this is the same meaning here. These are lipids that are not soluble in water, meaning they won't dissolve or dissociate or generally mix with water. Remember, we talked about bile when we learned about digestion. We said that bile is an important digestive secretion from the gallbladder that helps to emulsify fats or lipids. And so remember, we said emulsification isn't quite digestion yet. It's actually just breaking the fat molecule down into smaller parts. And so emulsification is important and necessary to happen before fat digestion, because emulsification starts to make the fat a little more accessible in our water-based bodies, accessible for the remaining processes of digestion. And then this word fat, we use this word when we're really specifically referring to fat that we find in our foods. So the word lipid, we're going to talk about three types of lipids, right? Remember, we're saying lipids are a large and diverse group of molecules. So we're going to talk about triglycerides, we're going to talk about phospholipids, and we're going to talk about sterols. Triglycerides, you'll see, are the most abundant fat, lipid in our bodies and in our diets. And so, or I should say specifically in our diets. So when we use this word fat to refer to lipids that are in our foods, we're often referring to triglycerides. So, so here you go. I said we'll talk about triglycerides, phospholipids, and sterols. So three types of lipids found in foods and in body cells. So when we're talking about lipids as they occur in food, that's really where we use that term fat. Again, you'll see I'm going to be using these terms interchangeably, and for our purposes, it's okay if you use them interchangeably too. So triglyceride, let's look here. So triglyceride is our first example of a lipid. So the triglyceride is this glycerol molecule bound to three fatty acids. So that's so here's glycerol and here's one fatty acid, but this is the actual triglyceride molecule down here. So you'll see we have one glycerol. This is the small schematic in the middle, or pictorial representation in the middle. So one glycerol, that's the backbone, and then these three wavy fatty acid chains that come off the side of the glycerol. So let me go back here. So triglyceride equals one glycerol molecule attached to three fatty acid molecules. Triglyceride is glycerol plus three fatty acids. So that's what we see here. One glycerol as the backbone. Sometimes we call it the glycerol backbone with three fatty acids. If you take a close look, and that's kind of what the picture up top is affording us the opportunity to do, you'll notice that the only elements that make up these molecules are, again, we saw this before when we classified our macronutrients. We have carbon, hydrogen, and oxygen. Those are the only three elements we have between glycerol and fatty acid. We said that's the same as we saw with carbohydrate, and as we'll see, we're going to see the same thing with protein, but protein has one little nuance to that role, which we'll get to in chapter 6 lecture. So you do need to know what makes up a triglyceride. When I've mentioned the concept of building blocks before, I've said that our macronutrients are large molecules made up of smaller molecules, and we can call those smaller molecules building blocks. These are the building blocks of triglyceride, your glycerol backbone and your 3 fatty acids. So I do want you to know that. All right, so what are triglycerides besides glycerol and fatty acids? Sorry, maybe I'm getting ahead of myself. First, let's go through the fatty acids in a little bit more detail, because in order to understand triglycerides, we have to actually understand these fatty acids. So when we give names to different triglycerides, because there isn't just one type of triglyceride, there are many different types of triglycerides, many of which you've probably heard the names of before, like we'll talk about saturated fats and unsaturated fats. These are types of different types of triglycerides, basically. So the name is based on the fatty acids themselves. So when we go back here, the glycerol backbone in a triglyceride is always going to be the same, this 3-carbon backbone. But these fatty acid chains that come off the glycerol backbone can be different. And they differ in a few ways. They differ in terms of how long the carbon chain is. Let me go back and show that. So let me use this image up top. So you'll see this is the fatty acid, right, in the yellow or the orangish. You'll see it's literally just a chain of carbohydrates. So the number of carbohydrates in this chain can vary from as few as 4 to up to the low 20s, so like 22, 24. So the number of carbons in the chain, that can determine the name of the triglyceride. Also saturation level, which we'll talk about. I've got some pictures coming up to help explain that. And then shape, which is influenced by saturation level. So you do need to know this as well. So let's talk about chain length again. As I said, the chain length can be, I said, as short as 4 carbon atoms. We call those short chain fatty acids. This term might be ringing a bell. I've said short chain fatty acids before. And let's see if you can remember where we talked about this. It was just last lecture in chapter 4, I said that the beneficial bacteria that live in our large intestine make short chain fatty acids as they digest our soluble fibers. So we've already talked about short chain fatty acids. They exist in our food too, but again, they are produced by some of those symbiotic bacteria that inhabit our colon, and those bacteria make short chain fatty acids from fibers that we eat. So by definition, short chain has to have fewer than 6 carbons in the chain. Then we have medium chain fatty acids, so it's going to be 6 to 12 carbons in the chain. And then long chain fatty acids, we're also going to spend a bit of time on long chain fatty acids. These have to have more than, or at least 14 carbons in the chain or more. We are also going to introduce some further nomenclature specific to chain length. So this nomenclature here is helping us understand, well, which end do we start counting from, right? If I go back here, which one of these carbons is carbon number 1 and which one is carbon number 12, right? Which end do you start with? So you'll see here actually, this is called a carboxyl group, and you see this Greek symbol alpha, and then we see this one is a methyl group, and you see that Greek symbol omega. And even if you don't know what alpha and omega mean, think about alpha, like what are some of the first thoughts that come to your mind? Potentially something towards the beginning, right? First or the beginning. So we actually begin our counting of the carbons in the chain at the omega end. And so the omega end, sorry, I'm saying that wrong, at the alpha end, the first carbon is the alpha carbon. Okay, so we start with the alpha end. The alpha end, or the carboxyl group, this thing highlighted in white, that's a carboxyl group. That's some basic organic chemistry. That's just the name of when you have a carbon double bound to an oxygen and then also bound to an alcohol group. We call that a carboxyl compound. So the carboxyl group contains the omega carbon, and the carboxyl group is always going to be the end that is attached to the glycerol backbone. So these right here are our alpha carbons. The alpha carbon is the one in the fatty acid chain. The carbon over here in the glycerol backbone, that doesn't get a number. So these carbons right here are the number 1 carbon in the chain. So understand where we begin the counting. We begin the counting at the carboxyl end, which is the alpha carbon, or the first carbon. And then omega is referring to last. So that's why they're given the names alpha and omega, because alpha means first, omega means last. And then the methyl end is the other end, the like free end of the fatty acid chain. And in white over here on the top right corner, that's a methyl group. A methyl group is always going to be a carbon with 1, 2, 3 hydrogens bound to it. So the methyl group is the omega end. Okay, so why is fatty acid chain length important? Because it determines how the lipid is digested. Pretty straightforward. The shorter the chain, the faster the digestion. Same with absorption. So the shorter the chain, the faster the digestion and the absorption. Also the faster the metabolism. And that then means there are different uses for these different types of triglycerides in the body. So short chain fatty acids can actually be readily used for energy. So can medium chain actually. And then we'll see one of the, that's not the only thing they can do, but that's common. And then we'll see that we're going to talk about some specific types of long chain fatty acids. And we'll see that one of the things they can do is actually act in the inflammatory pathways in the body. I'm just going to abbreviate that inflammation. Okay, then, so that's chain length. Then we said we can also classify the fatty acids in the triglyceride based on their saturation level. So we can talk about, or we can define saturated triglycerides or fatty acids, monounsaturated fatty acids, and polyunsaturated fatty acids. Let me go to this picture here. So I want to start with the saturated image. And let me, actually, I might go all the way back again. Forgive me, I'm going to go back here. So let me just erase a little bit, because now instead of talking about chain length and numbering, we're going to talk about saturation. So a real quick review of basic chemistry. You might remember that carbon atom has 4 potential binding sites. Oops, sorry about that. So remember, if we have a carbon, I don't know why it does that. Okay, here we go. If we have a carbon atom, that's a C, it can bind 4 other atoms, right? It has 4 potential binding sites. And so if we look at this up here at the top, at this fatty acid, if we look at all of these carbons in the chain, we'll see, oh, yeah, actually every single one of them is bound in 4 directions. This one here in the carboxyl group, it's not actually bound in 4 directions, but it still is sharing 4 bonds. It's just that with the oxygen, it's sharing a double bond, right? So this picture of this fatty acid chain up top here, this is actually a saturated fatty acid. The term saturated is referring to the number of hydrogens, or the saturation of hydrogens, or that the carbon atoms are saturated with the maximum number of hydrogens to which they could potentially bond. So what we see is, let's just look at some of these carbons in the middle, right? The carbon, let's look at this one. This carbon has to bind behind it and in front of it to another carbon to make the chain, that means it has two other binding sites available. So it's going to bind above and below with hydrogen. Same thing here, same thing here, 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 here. So each of these carbons in the chain is binding the maximum number of hydrogens possible. Therefore, all of these carbons in the chain are saturated with hydrogen. So when we look at this here, saturated fatty acids have hydrogen surrounding every carbon in the chain, meaning they're binding the maximum number of hydrogens, meaning there are no double bonds shared between carbon atoms. I'm just going to add that in here, between carbon atoms. Now, just for comparison's sake, let's talk about mono unsaturated. So again, let's break this word apart. Unsaturated meaning it's not saturated anymore, right? It's no longer saturated. And then this term mono meaning one. So a monounsaturated fatty acid is lacking one hydrogen atom, meaning therefore we're going to see one double bond. So the mono is referring to the one double bond, and the unsaturated is referring to the fact that the carbons in the chain are now no longer saturated with the maximum number of hydrogens they could bind. So in this schematic, we see the saturated one up top, and this is just a shorthand. There, instead of drawing the chain out like we saw, representing each individual hydrogen, this is a shorter hand way of writing it, and it's telling you that this carbon is bound to two hydrogens, this one is bound to two hydrogens, bound to two hydrogens, they're all bound to two hydrogens. So they're all saturated carbons. And then in the monounsaturated, we see the presence of one double bond. So that means that this carbon and this carbon are sharing two bonds between them. So they're sharing a double bond. And because of that, then they're each only bound to one hydrogen atom, where they could potentially each be bound to two hydrogen atoms. So I think this is actually, monounsaturated fatty acids lack, this should actually be two hydrogen atoms. You don't need to know that specifically. I more want you to know that monounsaturated have one double bond. But if it helps you to understand why the name, then you would see that it's missing two hydrogen atoms so that these two carbons can share the double bond. And then polyunsaturated, again, just building on to this word. So still unsaturated, meaning the carbons are not binding the maximum number of hydrogens that they can. But now instead of mono, we see poly. And remember, poly means many. So in this case, we should probably see many what? Many double bonds. And so let's see, we do. We see one and two. Well, we see at least a couple. So in polyunsaturated, in this case, it just means the presence of more than one double bond. So polyunsaturated fatty acids have more than one double bond between some of the carbon atoms in the chain. So then if you remember back in the beginning, I said we can also then talk about shape of these fatty acids. But I said shape is determined by saturation level. So here we see saturated fatty acids tend to be solid at room temperature. And this again, don't try to memorize this, but actually think about it. Think about it. So when we have this chain here with the maximum number of hydrogens, it's actually going to be a pretty solid molecule. It's going to be kind of rigid because there's so many hydrogens jam-packed into that space. So the other thing that happens then is these rigid fatty acids can actually stack on top of each other pretty nicely. See how these shapes are all the same? So we see saturated fats in wherever we see fatty foods that maintain a solid shape at room temperature. So butter, for example, obviously if it's a warm room, the butter will begin to melt a little bit. But lard, like if you cook bacon or other meats in a pan and then you don't wash the pan, and you'll see the fat will solidify after a short while once the pan cools off and cools back down to room temperature. So we see saturated fats primarily like animal fat, so meat and dairy. And then we do see small amounts of saturated fat in some plant foods. Coconut oil is the one that comes to mind. I think that's the most obvious example of a saturated plant food. Sorry, a plant food that contains saturated fat. If you ever buy coconut oil and keep it at your home, or even if you just look at it at the grocery store, you'll see that coconut oil is solid in the can, whereas something like olive oil is liquid. So olive oil has a lot of monounsaturated fatty acids in it and some polyunsaturated. Peanut oil has a lot of polyunsaturated and some monounsaturated. So again, think about this, don't just try to memorize it, but once you start to have these double bonds, you see that the shape of the molecule becomes less linear. It starts to be a little more zigzaggy. And so as we have these unsaturated fats, both mono and poly, we start to get all these varieties of shapes. And so these different shaped fatty acids with all these kinks from the double bonds won't fit together uniformly. So they won't stack and make a solid. So mono and polyunsaturated fatty acids, when we see these in foods, these are liquid at room temperature. So we do want you to know fatty acid saturation and how it determines shape. So again, just review, saturated fatty acids form straight rigid chains packed tightly together, so they are solid. Oh, it says it right here. Solid at room temperature. That's also important. We're talking about whether they're solid or liquid specifically at room temperature, not ever, because fats, the nature of fats as chemicals is that they will change shape depending on the temperature. So we're talking about whether they're solid or liquid at room temperature. So again, we find saturated fats in animal fats, butter, lard. If you don't know, lard and tallow, again, that's the rendered fat from animal meat, usually after you've cooked it, and you have the fat that might have cooked out of the meat or gotten left behind in the pan. That's fat from the animal. So primarily we see saturated fats in our animal foods. Fatty acid shape. So this is a fun, actually, I just think this is fun, helpful chart. Again, taken from your book, and I apologize, I took out the table number. But so you'll see, as I said, coconut oil contains quite a bit of saturated fat. Another thing we're not learning in this class is that there isn't just one type of saturated fat. There are lots of different types. So the type of saturated fat that's in coconut oil is not the same saturated fat that's in butter or milk or beef or egg or chicken or turkey. So that's really interesting, and that's what determines the healthfulness of these foods, not just the fact that there's saturated fat in it, but the type of the saturated fat. So again, coconut oil is rich in saturated fat, and then you'll see primarily our animal foods, some the fish a little bit, and then you'll see the abundance of mono and polyunsaturated fats. So walnuts having a really high amount of polyunsaturated. Remember I said olive oil, that's probably the one, oh, and safflower oil. Those two have the most monounsaturated fatty acids. So this, I think, again, I won't test you on this one actually, but well, I'll test you on knowing which foods are abundant in saturated fat versus unsaturated fat, but you don't need to know the relative percentages of the different types of triglycerides per food. I think this is just a nice FYI. Okay, more about shape. So again, unsaturated fatty acids have that kink in the chain at the site of the double bond, so they don't pack together tightly, therefore they maintain a liquid shape at room temperature. And so it's not written here, but again, you can see our unsaturated fat-rich foods tend to be seafood, fish, and then we're basically seeing a lot of nuts, and then oils, right? But what do these oils come from? Cashews, seeds, olives. I think it's helpful to remember the actual food itself, and then obviously if you derive oil from it, it's going to have the same fat profile. Canola is, there's no canola plant. This comes from a rapeseed plant. Not a nice name, but it's a rapeseed, so kind of like a grassy plant. Same with safflower. So these are seeds. Corn is technically a seed. So that's where we find unsaturated fats in fish, nuts, seeds, and then some of our fruits, technically, but olives being a unique fruit that contains a concentration of fat. Okay, so this starts a whole new conversation here. This bottom chunk, cis and trans. So we're saying that at the double bond, we have a double bond like this, and each of these is bound to one hydrogen. Each of these carbons is bound to one hydrogen. But the hydrogen could be either here, or the hydrogen could, like above, or the hydrogen could be below. So how do we give name to that? If we have a hydrogen above, well, sorry, let me just get it on here first. So hydrogen above and a hydrogen below. So we have these two terms, cis and trans. Cis and trans. Again, before we go further at all, just take a couple split seconds and think about the definition of these two words that might come to mind first. Trans, maybe meaning reverse or opposite, right? Not quite reverse, but not the same, like not together per se. So trans means that what I drew up here is actually trans, where the hydrogens are occurring on opposite sides of the carbon chain. So opposite sides of the carbon chain, referring to the placement of the hydrogen atoms. Cis means the same. So in a cis orientation, we're going to see, again, this is only talking about at the site of the double bond, we're going to see that the hydrogens are occurring on the same side of the chain. So they're either both going to be below or they're both going to be above. Okay, so let's talk more about that. So cis polyunsaturated fatty acid, we're going to have a kink like this, right? We're going to have a kink, basically. Let me just write that. And then here's the actual chemical drawing of it. So you have carbon, double bond carbon, the hydrogens are occurring on the same side of the chain. Trans, I'll switch my color up here. Trans, remember we said opposite. So let's look first at the chemical depiction. So we have carbon, double bond carbon, we have one hydrogen above, one hydrogen below, they're on opposite sides. So what that does to the shape of the fatty acid chain, a trans unsaturated fatty acid makes the unsaturated fatty acid linear or straight, right? So remember, we've been saying this whole time that unsaturated fatty acids are going to be kinked, they're going to be bent, they're going to be liquidy. Well, trans throws a wrench in that. Trans fats turn bendy, liquidy fat into a linear, straight, solid fat. And that's kind of precisely why trans fats are quite harmful to the body. So trans fats are not really, they occur very rarely in nature. Trans fats are actually like laboratory made fats. So trans fats, like margarine is the classic example of a trans fat. Trans fats, we started making these, I want to say back in the 70s, 60s, 70s, because we thought that saturated fats were bad for us. We now know that too much saturated fat is bad for us, but saturated fats by them, we do need to consume some saturated fats. They're not necessarily bad, they're only bad, they can be potentially harmful or unhealthy after we consume too much of them. So back in the 60s, 70s, we thought saturated fats were bad for us, but people loved butter so much. So some food scientists developed margarine from vegetable oils that had these kinked fatty acid chains, and they said, well, why don't we just figure out a way to make that kink go away? And so they did. They developed this process called hydrogenation, where they force hydrogens into the carbon chain, thereby forcing the orientation of the bond to become more linear. So hydrogenation takes a cis unsaturated fat and turns it into a trans, and thereby makes it more linear, more solid, so we can make up a food that's going to be solid in shape, but originally derived from something that was not a saturated fat. So this process again was developed to make oils take on a more solid shape in food. It also did help to reduce the rancidity. It kind of extended the shelf life, but the disadvantage is that we started to realize that these trans fats actually increase our risk for cardiovascular disease. So trans fats are highly recommended to avoid, just straight up avoid. They are not nutritious to the body. They are only a health risk factor. Okay, if you have questions on trans fats, definitely feel free to email me. You know, I could say a whole lot more, but that's kind of that's the extent to what we'll talk about in this class. Yeah, so whenever you see partially hydrogenated oil in an ingredients list, you know that it contains trans fat. You can also look on the nutrition facts panel. Trans fats have to be listed there by law. We didn't quite talk about the generally recognized as safe rule yet. We talk about that later in the semester, but there was a time, again, back before the 2000s, when we thought that trans fats were okay. So they became relatively prevalent in our food supply. But now we know they're not healthy for us, so they're not recognized as safe to be in our foods anymore. So it was by again, in the early 2000s, the FDA the Food and Drug Administration put out a ruling that the trans fats needed to come out of food and be completely out of food by 2018. So most manufactured foods, theoretically all of them, don't contain trans fats anymore, but trans fat is still on the label just so you can be sure of that. Okay, so now let's talk about essential fatty acids. We're still in this conversation of triglycerides, and in fact, we're still in this conversation of unsaturated fatty acids, and specifically poly. So we're talking polyunsaturated. Sorry, writing with the mouse is not my strong suit. So polyunsaturated fatty acids, I'm going to abbreviate that FA. So within this category of polyunsaturated fatty acids, we have what are called essential fatty acids. These are specific polyunsaturated fatty acids that our human bodies cannot make. Therefore, we must obtain them from our food. So again, just think of what the word essential means. They're essential to the body, but they're specifically essential to our diet. It is essential that we consume essential fatty acids in our food. So we're going to talk about two types of essential fatty acids, omega-6 fatty acids and omega-3 fatty acids. Specifically, we're going to talk about an omega-6 fatty acid called linoleic acid, and we're going to talk about an omega-3 fatty acid called alpha-linolenic acid. We find linoleic acid in vegetables and nuts, and we find alpha-linolenic acid in some dark green leafy vegetables specifically, as well as soybeans, flax seeds, walnuts, and it's not here, but also fish. So up here, I say that the essential fatty acids are precursors to these molecules called eicosanoids that regulate cell function. Specifically, what they regulate is cell function related to inflammation. So remember I said back in the beginning when we talked about chain length, I said that some of our longer chain fatty acids do some body regulation specific to inflammation. These are what I'm talking about. So these happen to be long chain fatty acids. These also happen to be polyunsaturated fatty acids. So let's talk more about them. So alpha-linolenic acid is abbreviated ALA. This is an omega-3 fatty acid, and when we ingest alpha-linolenic acid, our bodies will make from it these two other amino fatty acids, eicosapentaenoic acid, which we abbreviate EPA, and docosahexaenoic acid, which we abbreviate DHA. So all three of these, ALA, EPA, and DHA, are all omega-3 fatty acids. Okay, let me go back one more. So omega-3 fatty acid, this alpha-linolenic acid, we don't find that specifically in fish. I misspoke earlier. What we find are these two derivatives in fish. We find EPA and DHA in fish, shellfish. So we find some omega-3s in fish, but specifically we find EPA and DHA. So the reason this is important, that fish contain these two fatty acids, EPA and DHA, is because while our human bodies can make EPA and DHA, as far as we understand so far, our conversion, the human body's ability to convert alpha-linolenic acid to eicosapentaenoic and docosahexaenoic acid is not very efficient. So we haven't quite, the jury's somewhat still out as to whether or not the human body can make enough EPA and DHA from alpha-linolenic acid. There's plenty of science to suggest we can, and then there's also some science suggesting, well, just how much EPA and DHA do we need? So to be continued as nutrition continues to evolve. So EPA and DHA are precursors to eicosanoids. You don't necessarily need to know this word eicosanoid, but effectively I do want you to know that EPA and DHA promote growth and health. They help regulate the motility of our digestive tract, so they help facilitate healthy regular bowel movements, as well as just digestion itself. They regulate secretions of the digestive tract. They help to regulate blood clotting. Again, that's related to inflammation, and they regulate vascular permeability. Specifically, EPA and DHA will also stimulate prostaglandins and thromboxanes. You don't need to know those two words, but these are two eicosanoids technically that reduce the inflammatory response. So that's kind of the main takeaway that I think people often have with EPA and DHA, is that they reduce the inflammatory response. This is where we say these are anti-inflammatory compounds. And also, in part because of their anti-inflammatory nature, they protect our bodies against cardiovascular disease. So they're very, I guess we could say, heart-healthy fatty acids. That's EPA and DHA, eicosapentaenoic acid, and docosahexaenoic acid. Those are omega-3 fatty acids. So, oh no. Okay, I feel like my slides are getting mixed up. So let's look at the chemical structure of alpha-linolenic acid. And also this nomenclature, omega-3, right? If you remember when we talked about nomenclature in the beginning, we said omega means the end of the chain. So if this is the fatty acid chain right here, the end of the chain is the one with the methyl group. That's the omega end. So I also misspoke earlier. I'm so sorry. I guess something about recording these lectures on my own time. It's like my brain isn't with me always. I think I subtly misspoke earlier when I was naming the alpha end and the omega end. So alpha does mean first and omega does mean end or last, but we actually begin counting at the omega end. So whatever I said in the beginning, ignore that. We begin counting at the omega end. Anyway, I probably won't test you on this, but just so we get it straight, we begin counting at the omega end. So the first carbon in the chain is this one over here at the end of the chain. This is carbon 1, this is carbon 2, this is carbon 3, 4, 5, 6, 7, 8, and so on. So when we see an omega-3 fatty acid versus an omega-6 fatty acid, this is telling us something about the third carbon from the omega end, right? So something about the third carbon from the omega end. Well, that's interesting. What do you notice? The third carbon has a double bond. Okay, so that's what that means. Omega-3 fatty acids are, by definition, long chain fatty acids. You'll see this is an 18 carbon chain that has a double bond, its first double bond, occurring at the third carbon from the methyl end. So again, you don't really need to know this full nomenclature for my class. If you did take organic chemistry or if you ever do, you'll need to know a heck of a lot more than this. But if you're interested, I'll just explain the rest of what's on this page because I think it's actually quite interesting. So the full chemical name, not chemical, but the full name for alpha-linolenic acid, or I guess the abbreviated chemical name, it would be written 18 colon 3 omega-3. So this is telling us a lot of information right down. I'm down here at the bottom. So 18 is telling us that it's an 18 carbon chain. The 3 is telling us that there are 3 double bonds in the chain. And the omega-3 is telling us that the first double bond occurs at the third carbon from the omega end. So let's look at the full chain. Actually, let's look at this chain because it's showing the kinked nature of it. So we've got the omega end, we've got carbon 1, 2, 3. Okay, great. There's my first double bond, the third carbon from the omega end. And then if we keep going, we see one more double bond, that's 2. One more double bond, that's 3. And then if we keep going, we see this was an 18 carbon chain. So that's where you get this full nomenclature, 18, 3, omega-3. Yeah, okay. So now let's talk about, we're not going to talk about, oh dear. Alrighty. Apparently, I don't have a slide for linoleic acid, which I thought I did. That's okay. I have significantly less to say about linoleic acid. Linoleic acid is one of our most, in fact, the most abundant omega-6 fatty acid that we find in our foods. Is that true? That might be arachidonic acid. So let's say it's one of the most abundant omega-6 fatty acids that we find in our food. So really the only takeaway I want you to have with omega-6 fatty acids, I'm going to go back here. Omega-6 fatty acids are also precursors to eicosanoids, right? If I go back far enough, we will see that right here. Essential fatty acids cannot be made by the body. We have to eat them, and they are precursors to eicosanoids. All of them, both of them, both omega-6 fatty acids and omega-3 fatty acids. So linoleic acids are also precursors to eicosanoids, which regulate cell function. The difference is that omega-6 fatty acids tend to turn on inflammation, whereas omega-3 fatty acids tend to turn off inflammation, right? That's what we saw right here. The EPA and DHA, these omega-3s, reduce the inflammatory responses, whereas linoleic acid, this omega-6, tends to turn on inflammation. I'm not going to write that here with my mouse. I'm going to spare my hand, but please jot that down. I do hope that you will know that. And then just for the sake of redundancy, I guess, let's look at the nomenclature here for omega-6. So again, we'll start at the methyl end. This is going to be carbon number 1. And what did we say omega-3 meant? It meant something about the 3. It meant the first double bond occurred at the 3rd carbon from the methyl end. I feel like I just said omega end. Yeah, omega and methyl, right? Okay, great. Omega means omega and methyl are the same end. Okay, so in this case, then the omega-6 is going to see its first double bond at the 6th carbon from the omega end, or the methyl end. So let's check that out. Let's see if that's true. So this is carbon 1. Ooh, here we go. And we've got some shorthand here. So we've got, we actually have 4 carbons bunched right here. So we've got first carbon, and then we're going to have 2, 3, 4, and 5. And then this one is actually our 6th carbon. And we see it has a double bond. So omega-6 fatty acids have the first double bond occurring at the 6th carbon from the methyl end. And then the full chemical name for linoleic acid is 18,2 omega-6. So again, it's the same meaning. So it's an 18 carbons in the chain. And if you look up at this schematic, you'll see 1 goes all the way to 18 carbons. 2, meaning it has 2 double bonds. And again, let's use this one up top. So yep, here's the first one, and here's the second one. It's the only double bond between carbons. And then the omega-6 means that the first double bond is at the 6th carbon. We see that checks out. So those are your essential fatty acids, linoleic and alpha-linolenic acid. Now let's talk about phospholipids. So phospholipids, we can see this kind of a compound word. We have phospho and we have lipid. So this means we're going to have lipids attached to some sort of phosphorus compound. So phospholipids also contain the glycerol backbone, also contain 2 fatty acids, not 3, they contain 2 fatty acids, and instead of a 3rd fatty acid, they contain this phosphate compound. Phospholipids are unique in that part of them are soluble in water. It's this phosphate group that is water soluble. The fatty acids are still going to be water insoluble, same with glycerol. So because they're soluble in water, phospholipids actually can help us transporting fats in the bloodstream. Because remember, the blood is going to be water-based, so this phosphate head, we say, the phosphate head can float along nicely in the blood, and then the fatty acid tails can hold the fat. So that's a really important job of phospholipids. Remember when we've talked about bile? Bile is also composed of some phospholipids. Specifically, lecithins are phospholipids, and they are the component of bile that does the job of emulsification. And we make phospholipids, so we don't have to eat them. So here's your pictorial representation. So I'll start at the top. So here's glycerol. Right here, that's your glycerol backbone. Here's your fatty acid chain number 1, fatty acid chain number 2, and then instead of a 3rd fatty acid chain attached to the 3rd carbon here, we have this phosphate compound that has a long tail with it. So in the bottom, same thing. Here's your glycerol backbone in the middle. And you've got these 2 fatty acid chains, and you have this large phosphate head that's attached to the glycerol. So it wasn't in the list here, but another place we see phospholipids is in this phospholipid bilayer that is the cell membrane around every cell in your body. So you've got the phosphate head, that's the water hydrophilic, water-loving component, and then you've got these hydrophobic, water-not-loving tails in the middle. So that's why it's a bilayer, a double layer of phospholipids, because you have these water-soluble components facing out, and then the water-opposing components facing in. And that's also how it makes a nice barrier, is because the cytoplasm inside the cell and any of the extracellular fluid outside the cell are going to be water-based to make this nice, actually, fatty barrier around all of your cells. I guess that's all we're saying for phospholipids. So sterols is our third type of lipid. Sterols are unique in that they are rings. They're multiple rings of carbon rather than having the glycerol backbone and the fatty acid chain. So sterols are unique for that purpose. Plant sterols appear to block the absorption of dietary cholesterol, so that's helpful. Okay. And cholesterol, we have it here because cholesterol is a sterol. Cholesterol is a type of sterol. So we only find cholesterol in animals like you and me. We make cholesterol, humans make cholesterol, as do any other animal that we might eat. All human cells can make cholesterol, although most cholesterol is produced in the liver. Because of this, cholesterol is not a required nutrient. We make as much cholesterol as we need. We use cholesterol to make hormones. Our reproductive hormones are cholesterol-based. Vitamin D is cholesterol-based. And bile, some of the components in bile are also cholesterol-based. So sterols are really interesting, I think. So let's take a look at the chemical depiction. So here on number A, letter A is your basic sterol ring structure. And then an example, cholesterol. You can see it has the ring backbone, or kind of the ring base, let me call it base, and then it will have some sort of attachment to it. That's what makes cholesterol. And then a cholesterol ester, I skipped over it here, but we said that as cholesterol kind of floats around in our blood, we're going to see it existing as cholesterol ester or cholesterol esters. So cholesterol ester just has this extra fatty acid chain attached to the cholesterol molecule. Okay, so let me just go back then and highlight what I want you to know about cholesterol, sterols. So I want you to know that plants make sterols, but they don't make cholesterol. So the only place we find cholesterol in our food is when we eat animal foods. I also want you to know that humans make cholesterol, so we do not need to eat cholesterol. That's why eating cholesterol can be so harmful, is because we're adding a bunch of cholesterol when we already have enough. I want you to know that. And then I want you to know what we use cholesterol for. I want you to know these three things. So we use cholesterol to make some of our hormones, specifically our reproductive or sex hormones. We use cholesterol to make vitamin D, and we use cholesterol to make some of the components in our bile. Reproductive hormones, if that's not ringing a bell, I'm talking about our estrogens, I'm talking about testosterone, I'm talking about progesterone. Yeah. Okay, now digestion of fat. I'll go through digestion of fat and then I'll pause for this lecture, and then I'll pick up for part 2. So fat digestion, remember we said begins in the small intestine. Begins in the small intestine, yeah. We have cholecystokinin and secretin. These are two of our digestive hormones that trigger the gallbladder to release bile. Remember, bile is produced by the liver, stored in the gallbladder, secreted into the duodenum of the small intestine, and bile's job is to emulsify fat into smaller droplets so that then the pancreatic lipases, which are enzymes, can actually digest fat down into 2 fatty acids, and basically the glycerol backbone attached to 1 fatty acid. So we call that a monoglyceride. Mono meaning 1 fatty acid attached to the glycerol backbone. I do have a picture of that. Okay, let's say all that again, just because now we have a picture. We did say that there may be tiny bits of fat digestion in the mouth, because we have lingual lipase. Little lipid digestion occurs here. The stomach, We do have gastric lipase as well that will digest triglycerides a little bit, but really, for the most part, fat digestion occurs in the small intestine. So liver produces bile, gallbladder releases bile into the duodenum of the small intestine. Here's your triglyceride, your glycerol backbone, and your free fatty acids. And we said, well, the pancreatic lipases will break that down into a monoacylglyceride or a monoglyceride. That's this. The glycerol backbone attached to one fatty acid, and then your other two fatty acids now dissociated or separated. We'll also see this is a cholesterol ester, remember? So when we eat cholesterol from our food, it'll often come in the form of cholesterol esters. So as we digest those, we're going to just break those apart into cholesterol and again, just a free fatty acid. And if we do have phospholipids coming in, if we do ingest any phospholipids, we're also going to break those apart into the phosphate head and then two fatty acid backbones. And now here's where fat digestion gets a little bit complex. So all of these components, let me use a different color. All of these digested components are going to get packaged into something called a micelle. So we see here in the schematic over here in the lumen of the small intestine, these micelles are going to form. So these are the products of fat digestion. All these things right here in the orange circle are going to combine with a smaller amount of bile salts to make these cool little like fat packages. Because again, fat doesn't mix with water. Our bodies are water. So fat has to have this extra way of getting absorbed and transported. So the micelles get absorbed into the enterocyte, and then a whole other unpackaging and repackaging happens. So you'll see this is showing that short chain fatty acids can get directly absorbed into the blood. And you'll see this other word here, Kylo Micron. Use green. So Kylo Micron. So this is this new packaging of the fat components. The chylomicron is made in the enterocyte. So within the enterocytes, components from the micelles basically get unpackaged and then repackaged as chylomicrons. And the chylomicron gets absorbed into the lacteal, which is your lymphatic system. And then eventually the lymph will drain into the blood so that these different fat components can get delivered to different sites of your body as needed. Okay, so let's say all of that again in larger font. So absorption of fats absorbed in the small intestine. We saw minute amounts of digestion in the mouth and the stomach. The micelle is the lipid digestion products that get absorbed into the enterocyte. Chylomicrons are lipoproteins that get packaged in the enterocyte and then get sent out into the bloodstream. So chylomicron is basically a package of lipoproteins. So again, just look at what that is. Lipo meaning lipid, protein meaning protein. So these are, chylomicrons are these like large protein and fat globules. And that's how we transport fats through the body. So a lipoprotein is fatty acids surrounded by phospholipids and proteins. Proteins are water soluble and phospholipids are water soluble. Fatty acids are not, but because we have the phospholipids in the protein making this chylomicron package, we can transport this in the blood. Chylomicrons get transported out of the enterocyte. They'll go through the lymph system first, and then they'll get transferred to the bloodstream at the thoracic duct. You don't really need to know that, but again, if you've taken anatomy and physiology, you might recall the thoracic duct from the lymph system. We said that briefly, I just said that short chain fatty acids can get absorbed quickly. Same with our medium chain fatty acids. So those will get absorbed directly into the blood because they are so short and simple. I remember way back in the beginning of this lecture, I said that the length of the chain of the fatty acid will determine the digestion, the absorption, and the transportation through the body, the metabolism. So this is that. The short and the medium chain fatty acids get absorbed quickly. They get absorbed directly into the blood. They don't go through the lymph first, and they are generally metabolized more quickly. And they are not in the chylomicrons. Triglycerides in the chylomicrons must be disassembled by lipoprotein lipase before they can enter the body cells. Again, just another enzyme, but in this case, it's an enzyme that's specific to lipoproteins. So it's a lipase, so it can digest fats, but it's a lipoprotein lipase, so it can specifically digest lipoproteins. And then once the triglycerides get into the body cells, what do we do with them? They can be a source of energy. We can use them to make other lipid-containing compounds that we might need to make. And we can also store triglycerides in our muscle and in our adipose as triglyceride, as fat. So that might seem a little wonky, but it's not crazy. We do store small amounts of fat in our muscles. And in fact, some endurance athletes, because remember we saw that slower, longer duration, well, we haven't quite seen that yet because we'll talk about physical activity later on, but lower intensity, longer duration activities like hiking or marathon running are going to use quite a bit of fat over the duration of that exercise, I guess. And so people or athletes who are really trained in kind of lower intensity, longer duration exercises do actually have quite a bit of intramuscular fat, so fat that is stored in the muscles. All right, so going back to the small intestine, we've talked about bile salts and the fact, their job basically, that they emulsify fats or lipids or triglycerides. But here's the first time we're actually seeing a picture of fat emulsification. So here's your large fat droplet. You see all these triglyceride molecules in this large fat. And here you see the bile salts on the outside. I see it's got some phospholipids in it, right? Some lecithins. And then you'll see the bile salts basically just make smaller fat droplets. And then what this is showing is that the pancreatic lipases, the digestive enzymes, can now actually work on the small fat droplets. They couldn't work, and here you'll see this pancreatic lipase is digesting this triglyceride, making the monoacyl glycerol, the monoglyceride, and the two fatty acid chains. So here again is your chemical representation of the triglyceride being digested by the pancreatic lipase. So you've got the glycerol backbone and the three fatty acid chains. You'll see the lipase is going to break these bonds between, well, it's only going to break two of them, right? Because we're going to wind up with a monoglyceride. So you'll see it's going to break this bond that attaches the carboxyl carbon to the glycerol carbon. Is going to break this bond, and so you're going to wind up with two free fatty acids and one monoacyl glyceride or monoglyceride. Sorry. And then just a quick little review from chapter 3. I think we talked about a few of the chemical reactions of digestion. See if you can remember the chemical reaction in which we apply water, or water is added or used to break something apart. What type of reaction is that? Break something apart is lysis, and if there's water involved, maybe hydro, so hydrolysis. Alrighty, and then so we make these monoacyl glycerides, we make these free fatty acids. Now we're going to package them up into the micelle. So here you see free fatty acid, monoacyl glyceride. Well, remember we've got some cholesterol esters, and then we also have some further digested cholesterol. This is inside the lumen of the small intestine. So all of these things get packaged up into this nice micelle. The micelle gets absorbed into the enterocyte, and then, as we said, they just disassemble again so that they can be packaged into the chylomicron. So here's the chylomicron. So all those same components just package themselves right back up into the chylomicron. Remember, we said the chylomicron has this like outer membrane that is protein and phospholipid. So this is water soluble, so it can travel in the bloodstream. It'll go into the lymph first and then enter the bloodstream at the thoracic duct. So you'll see chylomicrons are full of triglycerides and cholesterols and lipoproteins and proteins and phospholipids. So again, so here's the full overview. Here's the gut lumen, here's your enterocyte, this is the enterocyte cell. The micelle gets absorbed into the enterocyte. The short and medium chain fatty acids can then go straight into your blood circulation. And we'll revisit albumin, but albumin is a little transport protein that helps get the short chain fatty acids from the enterocyte to the blood. And then as the micelle dissociates, we've got, again, all these components of fat digestion: monoglycerides, free fatty acids, cholesterol, phospholipids, glycerol. So they're all going to get packaged by the endoplasmic reticulum. They're all going to get packaged into this chylomicron. The chylomicron is going to travel into the lymphatic system and then eventually into the blood. And then again, what are we going to do with triglycerides? We can store them in adipose cells. We can store them in muscle tissue. And we store them because they are a source of energy. Remember, we said carbohydrate, protein, and lipid. These are macronutrients for a few reasons. They're macronutrients because they can be a source of energy. They're also large molecules made up of smaller molecules. They all contain carbon, hydrogen, and oxygen. So this shouldn't be new. This shouldn't be a shocker. Fats are a source of energy, right? Triglyceride is specifically the type of lipid that we use to supply energy. We don't really use phospholipids or sterols as energy. We really only use triglycerides as energy. And because we can use it as energy, it means we will want to store it. So any extra triglyceride we consume that we don't need immediately, we will store it in muscle and we will store it in adipose. Here's a picture of a fat cell. You see it has all of the nice triglyceride and then a small nucleus. Not a small one, but a nucleus that gets kind of pushed to the side. Let me see here. So I think I'll pause here. This is probably roughly halfway. So we'll pick up with the roles of fat in part 2 of chapter 5. All right, thanks for listening.